the hornets were like hanging out on the trees, just kind of going around hawking bees and grabbing bees just as they're like flying in to land at the hive.
We're at least fortunate that I think our local beekeepers have semi-automatic weapons still that they could use to shoot
That's
them.
probably about the right kind of size and shape of artillery you need, I think, for some of them. They really are big.
In this episode, I interviewed Dr. Brent Sinclair from the University of Western Ontario. That's located in London, Ontario, in Canada. He's currently a research scientist there, has over 190 research items published, 31,172 reads of his works, and 6,652 citations of his work. He is very well known for his cold tolerance work and good sense of humor. He was actually one of my initial inspirations to go into the sciences and study insects. So without further ado, here it is, my interview with Dr. Brent Sinclair.
Hey, how are you doing?
Tell me it's my best side. Good. Especially when I'm muted, of course.
I'm sorry that it looks like I've caught you in the field right now.
Well, I've been loving the virtual background, so I've been trying to like zoom from a different location for every day, which was kind of fun at first, but like, you know, it's starting to be like, oh, I've been doing this for 8 weeks now. Now it's just like added pressure to my day, you know.
Like, shoot, I need to pick another location before this next meeting.
Thanks.
Listen, you did your PhD at University of Otago. Saying it seems like a very traditional, rich university there.
Yeah, so I think it's the oldest university in New Zealand.
Oh, okay.
And it's a small town, and also Dunedin got rich because there was a gold rush in New Zealand in the 1860s, 1870s. And Dunedin got rich not because any like sizable amount of gold was ever found, but like all good gold mining things, they got rich out of being like providing the merchant sort of stuff to go with the gold rush. So similar to like San Francisco, right?
Okay.
And so they have this like university, you know, and it's got like big old buildings and so forth and such like. And there's lots of like big old houses from that era through the city as well. But the other cool thing about Dunedin, which you don't get in many places, is like it's a long way south for the southern hemisphere. So it's at 47 degrees south. So it's quite a long way south, and okay. It's got like just all of this wildlife all around. So it's built out on kind of a peninsula. There's the world's only mainland albatross colony, like on that peninsula. There's you know, so we had like, so as undergrads, like, you know, for our like conservation biology or conservation ecology third year class, you know, we'd have like field trips out to see, you know, albatross and penguins and seals, and you know, we'll like go and handle them, you know.
Oh, that's pretty nice. Yeah,
and they were like, you know, so as an undergrad, you know, there were plenty of people doing research, so you know, I would be able to volunteer and just go out and, you know, band seabirds or whatever, and you know, just like on the weekend or sometimes even just on like, you know, evenings or afternoons during the week. So it's a really cool place to kind of be immersed in wildlife.
Yeah, and there you did your PhD studying ecology and physiology of Alpine and Antarctic arthropods. And why, I mean, first off, you know, what is Alpine when we're talking about that, and why was that interesting? In
New Zealand, because it's a long way south and all those things, so the Alpine zone starts at kind of about 1,300 meters above sea level. And so there was actually a location called the Rock and Pillar Range, which is 2 hours drive from the department in a 4-wheel drive, where you could actually drive up into the Alpine zone. And so there it looks like Alpine tundra. It looks like Arctic tundra. So short little things, mostly flightless insects, really kind of like actual genuine Alpine conditions, and you know, cold and snowy and brutal during the winter.
Okay. And then so why was it interesting to study that? What was your particular interest?
I moved from Auckland to Otago in the start of my third year because I wanted to work on I wanted to do whole organism stuff, right? So I wanted to study like, you know, behavior and things like that. And I'd done a field trip to an island where we had like wandered around in the rain, you know, measuring bird behavior. And I was like, this is great. And then after a while, it turned out that actually what was great was the bit where you're out wandering around in the rain more so than the, because I grew up in like the suburbs and didn't really do any hiking or anything like that. So it was sort of the wandering around in the rain more than the bird behavior that turned out to be most interesting. And so I knew that I wanted to do stuff that was sort of field related and in interesting places.
Yeah.
And I knew that I was interested in insects. I'd had a summer job where I'd been out to a subtropical island helping to measure vegetation communities. And there were all these, because there were relatively few predators, introduced predators there were all these like amazing things, these like giant weta, which are the heaviest insects in the world. And
neat.
Like cool giraffe beetles. So they're they're related to weevils.
Okay.
And they've got these like super long weird noses and things. And they're in the family Brentidae.
Oh, wow. That's awesome. Did you name them Brentidae?
Oh, I always claim that I'm named after. Anyway, and so I thought that I might want to work on insects, but we didn't really have many insecty people. And in my third year of undergrad, I was doing this course called Environmental Physiology, Zoology 313. Still remember it. It's still called Zoology 313. Amazing.
Wow.
Anyway, and it had these guest lectures from this guy that was visiting the department on sabbatical, a guy called Bill Block, who worked at the time at the British Antarctic Survey. And he gave these 2 lectures on insect cold tolerance strategies. So insects that could survive freezing and insects that avoided freezing. And I was like, that is what I want to do. And I've basically been doing it ever since. I was able to go and sign up into the lab and sign up for an honors project with them, and got to go into the mountains a couple of times with Bill, and there was another guy visiting as well, Hans Ramlov, who's now a professor at Roskilde University in Denmark. I started working on these abundant Alpine cockroaches with the resplendent name of Celatoblatta quinquamaculata.
Oh, and beautiful name. Yeah.
Beautiful
Rolls
name.
off the tongue.
Yep, exactly. That would be in the exam. And they can survive freezing. You cool them down and they freeze solid and thaw out again and survive. And I was like, this is amazing, and I want to know more about this.
Yeah.
So that's kind of where the Alpine stuff came in. And then the Antarctic stuff and staying for a PhD really came about kind of simultaneously. So when you grow up in New Zealand, because it's the stopping off point for a lot of Antarctic research, including the US Antarctic program. But also it was the stopping off point for Scott and Shackleton and all those people when they were going to the Antarctic. So Antarctica kind of looms large in the psychology of the nation, if you like. And so, so I was like, how do I get to Antarctica? And and my to-be PhD advisor, David Wharton, did research on Antarctic nematodes. And he's like, well, we just basically write an application to go. And, if you wanted to stay and do a PhD, you could do that. And I'm like, sign me up.
Wow.
So basically, essentially, my main motivation was that here was an opportunity to go to Antarctica, which is a perfectly reasonable reason to stay somewhere and do a PhD.
Do a PhD. Yeah, of course. It's kind of funny because, the last person I spoke to, so Dr. Jenny Corey, she also started by studying bird behavior. Okay. And then her path to entomology was also rather serendipitous, it was just kind of following interest after interest and led in that same path. And I think with you, it seems kind of like similar, it wasn't, when you were born, you weren't like, I want to be an entomologist. It was kind of the opportunities arose and you just kept kind of pursuing them. Yeah,
I think there's kind of, there's 2 kinds of people in the world, people that think there's 2 kinds of people and people that don't. No, I think there's sort of 3 kinds of entomologists. There's the people that were like keeping bugs under their beds and had a butterfly collection at the age of 4, and right. And that was always what they were going to do. And there's people like me that kind of got into it fairly early on because it was like, wow, these are cool. And, sort of for intellectual reasons, there's neat things to do and they're neat animals. And there's people that I think just kind of fall into it because, they're looking for a research project and they want to study behavior or heat shock proteins or, or human disease or whatever, and then one day look back and discover that all they do now is work on locusts or something. So, I think there's sort of those three-ish categories. And I can see that even with the people in my lab. There's people that are just like, this is amazing, this is what I want to do, or this is always what I wanted to do. And people that are like, well, actually, like I see myself as something else, and this just is a system to do it in.
Right, yeah. And then you went on after your PhD to Space Lab, where you did a postdoc. This is in Stellenbosch University in South Africa. And that's,
so is spatial, physiological, and conservation ecology. Yeah.
Oh, it's not interstellar travel. It's
terrible false advertising.
Yeah, you're like, wait, what am I doing here?
I know. I didn't get launched on a single rocket the whole time I was there.
Yeah. Now, fortunately, there are some really neat, it looks like mountain ranges right behind the university.
Yeah. So this was, again, a bit of an accident. So at the time, and this is like one of these things that looking back, like in a different world, I would have interacted with a very different set of people. So because of David's interests and the people that I was exposed to, the intellectual world I interacted with as a grad student was really focused on kind of insect environmental physiology and specifically kind of Antarctic stuff. So the meetings I went to tended to be Antarctic meetings with other people that did Antarctic research and things like that. And and whereas with my students, I tend to take them to SICB or to Canadian Society of Zoologists or to things that have a very different emphasis.
Yeah.
So those were the people I knew. And so Stephen Chown was like kind of, and still is like just an absolute global leader in that sort of world, right? Invasive species, insect environmental physiology, polar biology of all stripes. And so he was just a clearly obvious person to go and work with.
Yeah.
And at the time that we kind of arranged all this, he was based at the University of Pretoria, which is just near Johannesburg. And he actually moved to Stellenbosch like a week before I arrived for my postdoc, you see. So it was just like, oh, okay, I guess we're going to Stellenbosch.
Yeah. So you weren't planning. You were originally not going to be in Stellenbosch.
No, not at all. Not at all. And it was just, how it worked out, right? So we had kind of the main field sites I had one in the Cederberg, which is this mountain range north of, in the northern part of the Western Cape, or north of Cape Town and stuff. So it gets cold at night. So, even in kind of October, which is like late spring, we were up there at kind of 2,000 meters, and there was ice on everything, and animals were frozen.
Oh my gosh!
Freezing and stuff, which was just really cool.
Oh,
and these areas are amazing because the Karoo Desert has such clear skies, and it's at high elevation that that's where the South African Large Telescope is based. So, you can imagine being up on this mountain range at 2,000 meters. We were staying in this I'll call it a hut. It was really kind of just a shack. Like it didn't even have a door. I don't think. And you could get up in the night and you could look down to the horizon, and there was just like the best stars I've ever seen, right down to the horizon, just stunning.
Wow.
So we worked on these amazing cockroaches there, these like big cockroaches, and these cool beetles of various sorts. And then we looked at some of the same cockroaches in the Karoo Desert, where it also gets very cold at night, down and also in the Drakensberg Mountains in Lesotho, or on the Lesotho South Africa border on the eastern side of the continent.
Okay.
And that had all sorts of interesting things as well, because I could fly across to Durban and drive up there, but I had all sorts of interesting things crossing the border, and I broke four-wheel drive vehicles and had to hang out with the police at the border station for a day and things like that.
I guess that all kind of helped give you the ability to navigate those types of countries and scenarios. Now, I mean, you're not intimidated, I guess, if you go there.
Yeah, well, it's interesting because I don't do that very much now for my research. Actually, the other thing I did from South Africa, which is possibly actually the kind of here's the point where I realized how challenging this was. I did a couple of trips back to the Antarctic. So I did 2 trips to Marion Island, which is 2,000 miles south of Madagascar, I guess is the best way to find it.
It's like in the middle of the ocean, right? I mean,
it's I think slightly less isolated than Hawaii, but otherwise kind of, as isolated. And it has a population, when they're doing the kind of the takeover, when the ship visits once a year for a month or so, it has a population of about 80. And then the rest of the time it has a population of like 12.
I remember, I almost went there for my master's. I would have been there for about a year.
Yeah.
I remember we had discussed that.
Yes.
And I was thinking, could I live on an island for a year with 7 other people? That was one of the things I had to think through and process. And I was like, that's...
Well, knowing you, Irfan, I think you totally could. I think
I could. I mean, yeah. I think it would have been a lot of fun. Yeah, absolutely. Yeah. But yeah, so tell me a little bit more about that.
So that was interesting. So that was it was the last days of the old research station there. It's now been replaced with a shiny new one.
Oh, nice.
But essentially, that was nice because there was a pretty well-equipped lab. So we could go there and there's only 50 species of insects on the island or something. So you could so Stephen Chan had been working there since he was a whippersnapper, since he was like a master's student, maybe even before that.
Wow.
And so,
cool.
You could just kind of say like, well, Stephen, I'm thinking about doing an experiment with something that does this and this. And, what do you think? And he'd be like, oh, like at this time of year, there's this species of weevil. You go to rocks with that species of lichen and, and on a wet day, you'll be able to pick them off with forceps or something like that. It was just amazing.
He like fully understood the ecosystem there and basically like,
it was just great. And
yeah.
So that was super fun, super hard work. That was like 24/7, like, because you're only there for a short period of time. So it'd have like all these experiments set up and
knock it all out.
Constantly hangry and, generally just kind of like challenged. And the second time I went down there, I designed this colossal experiment to like do this repeated freezing experiment on caterpillars. And what we hadn't quite figured out, what I hadn't quite factored in, was that it took me almost 12 hours just to set up the experiment for these things that were supposed to be frozen every day.
No.
So every day it was like, okay, 12 hours of kind of like crazily doing this, which is kind of crazy. But yeah. So there were all sorts of things like that. And I think you learn. And then the other trip that we did, so myself and 3 others, so John Teblanche, who's now a professor at University of Stellenbosch, Matt Scott, who is now a researcher at Scions, and Jaco Klock, who's now a researcher at Sable Systems in Las Vegas. We did some work in Antarctica for 3 and a bit months in this place called Cape Hallett, which is sort of quite well traveled now because they had a sort of a semi-permanent research presence there for a few years and things. But at the time that we were there, we spent the longest there that anyone had since the 60s, I think.
Oh, wow.
So there were just 4 of us living in tents doing physiology with generators.
Oh my gosh.
And that was challenging on all sorts of fronts. Our generators
are loud.
Generators stopped working.
Oh no.
And our radio stopped working after about 6 weeks, so we couldn't really communicate with anybody. So we're just kind of checking dates off on a calendar and kind of, on the day that it seemed like a ship was supposed to show up, we kind of like looking up and waiting to see if a ship showed up. So
it's kind of fun because that kind of makes you very adaptable to things that are out of your hands, things that are out of control.
Yeah. I don't know if it's made me personally any more adaptable. I still freak out when I'm still a control freak, and I still freak out when I'm not in control of stuff. But I think that it has made me very scientifically adaptable. And it's been quite interesting looking at that with this pandemic, with lots of people taking stuff home and maybe thinking about trying to do their science at home and stuff. And it's been a good thing to draw from. So I co-supervise a master's student in New Zealand, and we'd just sent a bunch of equipment to her, and she kind of took that home and all the animals. And I'm like, okay, so this is actually exactly the same as being stuck in a tent in Antarctica, except that you can go to the grocery store.
Right.
Yeah. So it's been quite useful for kind of like thinking through some of those things and in general, like adapting to the things that you get. And so I still do occasionally get to do field work in the Arctic, and those are skills that we retain, although very much try to do it right now, trying not to do it based out of tents with generators and things, just because you always get crappier data.
And then you went to go to, you did your postdoc studying stress genomics at University of Nevada.
Yep.
And there you studied Drosophila.
In Las Vegas.
In Las Vegas.
The last place in the world I ever thought I'd live. I'm studying Drosophila, the last insect, Drosophila melanogaster, the last insect in the world I ever thought I'd work on.
Fruit flies. That's funny. Yeah. And you're studying cryopreservation of fruit flies.
When I was first in Stellenbosch, like early on, I got an interview for a faculty position. And so I was like, this is sweet. I'm going to be able to get a faculty position after I finish this postdoc. No problems at all. It'll be great. And so then the next year I applied for like all the faculty positions.
Yeah.
And got precisely no interviews. And
wow.
Holy crap, what do I do, right? Because, I'm living in a country that, my visa is about to expire. There's no job for me back in New Zealand. And like, what do I do? So I emailed a bunch of people, and someone was like, well, Steve Roberts has a bunch of money available right now. He was at the University of Nevada, Las Vegas at the time. And it was sort of the golden, just before the economic collapse, when Vegas and Nevada had just all this money. And so he had money as part of a kind of a genomics core facility to hire a postdoc. So I kind of showed up and knew nothing about any of these things, and learned from him and from another faculty member who moved there just after I arrived, Alan Gibbs, how to wrangle flies, about fly environmental physiology and that sort of thing, and did some experiments on cold. And as I was doing this, I got interested in so I'd always been interested in evolutionary physiology, but there's this, basically all fly people have worm envy. They're all envious of C. elegans, because you can freeze C. elegans. You can basically like get it into the right life stage, pop in some glycerol and chuck it in the freezer and go on vacation. Whereas with Drosophila, you have to keep kind of, managing the stocks. So everyone wants to have those famous, C. elegans vacations. I don't know.
And that's your C. elegans vacations, and that's nematodes, right? So
yeah. So anyway, and so we wrote a grant to figure out if there were going to be any Drosophila species that were freeze tolerant, and then and the argument would be that either you figure out how they do it and make melanogaster do it. Which actually, like, down the line, my colleague Vlad Koshel has sort of figured out how to do.
Whoa.
But also, like, maybe there's just a species that we should be using as a model instead, right?
Which brings us to, afterwards, so then you become a faculty at University of Western Ontario since August of 2006.
Yep.
So that was, I guess, actually not too long after I started my undergraduate there.
I know you were there like pretty close to the beginning. So so I think many of the people so I think that so Heath McMillan, I think, joined my lab as a volunteer like 2 months after I started.
Whoa. Oh, neat. Yeah. Okay.
Yeah, so that was still quite early days.
Yeah, and he's a faculty now. Where is he?
Carleton University.
Okay.
Ottawa. Yeah.
Yeah. Oh, nice.
Yeah, so and I moved to Canada, and and it's been great ever since. About the same time that I arrived in Canada, and this is completely coincidental, I promise, emerald ash borer turned up in London, Ontario.
Oh, so that's a devastating borer pest. Anyone who's up in the Northeast is very familiar. We just got our first case of emerald ash borer confirmed probably about 3 or 4 months ago down here in Texas. We don't have a whole lot of ash, but it's a devastating pest of all ash. Yeah. So sorry,
exactly.
Go ahead.
So, insert, standard newspaper headlines of kiss your ash goodbye and so forth. Right.
Right.
So I'd been kind of in touch with a guy, Peter de Groot at the Canadian Forest Service, and just kind of said, hey, I'd like to do applied research, but I'm not a real entomologist. I don't even really know where to start. But it seems like, given that I work on cold and, and there's all these invasive species coming. Like, an obvious question is, will this thing survive in Canada? And he was like, oh, well, if I hear of anything, I'll let you know. And so then, of course, emerald ash borer comes through, and it was an obvious thing to kind of, to sink our teeth into. It was in the city. At the time, we didn't know how far it was spreading and how far it had spread already.
Yeah.
So when I first joined this thing, we'd go to like meetings that explicitly didn't have emerald ash borer written on the side of the conference room. You had to sign a secrecy agreement when you went in, but less people knew that, where things were. And, and then of course it turned out that it was everywhere and had been for a decade. And that sort of led to now almost half the work we do in the lab is applied in one way or another, which is kind of, insert new pest here. Will it survive in Canada? And we've worked on emerald ash borer. We've worked on spotted winged Drosophila Suzuki'i. We've worked on we've worked on brown marmorated stink bug. We're working on Asian longhorn beetle. And it's just kind of like one nasty after another. In the news today, the next thing that I'm going to do is sit down and edit like a little press release about these, like, the Asian hornets that are suddenly in the news as well.
Yeah, I was going to say a giant. You're gonna try tackling the giant hornet next.
So I had a postdoc who visited for a year from China, and we went out to visit him, and he's in like a bee science college, right, in Fuzhou in southeastern China, and I saw these hornets, and they're just amazing because they're colossal, right? They're like hummingbirds.
Gosh.
And we saw them, and what they were doing was they just kind of hang out. So these were like bee hives. So they try to have a hole that is too small that excludes the hornet from getting in, right?
Oh, okay.
But the hornets were like hanging out on the trees, just kind of going around hawking bees and grabbing bees just as they're like flying in to land at the hive, and then kind of going off and like chewing their heads off up the tree, which was just amazing. Unfortunately, it was the wrong time of year to see the Asian honeybees, Apis cerana, where they do this thing where they like surround them in a ball and then heat them up and kill them and cook them.
Yeah.
I was very disappointed not to see that, but yeah.
Oh, that would have been really neat. Yeah, I understand. I don't know that that honeybee is very common here in North America.
It's not. So it seems like the trade-offs are because Xinjiang does quite a lot of work on this Asian honeybee.
Okay.
And it seems like the issues are that it's much more resistant to diseases and stuff. I think many of which come from Apis cerana to Apis mellifera, right? Or come from Asia. But it's not as productive. And I think it's like the management is a little bit trickier.
Okay.
And they definitely look different. We would see them, and we see the wild colonies as well in the rocks and stuff when we're out hiking and things.
Oh, interesting.
Yeah.
Huh. Yeah, it'll be interesting to see kind of how that unfolds if they actually, the giant hornet actually establishes here, and what that means for a lot of our beekeepers.
Well, it's certainly another thing to worry about, right?
Yeah.
Yeah, I don't know if you've come across Gard Otis at Guelph, but he was doing some work in Southeast Asia, I think in Indonesia or Thailand, on these giant hornets and the way that the local beekeepers kind of deal with them. It's kind of cool.
Wow. See, I'd imagine. See, we're at least fortunate that I think our local beekeepers have semi-automatic weapons still that they could use to shoot.
That's probably about the right kind of size and shape of artillery you need, I think, for some of them. They really are.
So your lab now focuses on understanding the impacts of temperature extremes, high and low, on insect biology, ecosystems, and evolution. So why, in general, like an elevator talk, why is that important?
Okay, so insects are ectotherms. So when it's cold, they cool down and slow down and run the risk of freezing when it's below zero. Yet we find them everywhere it's cold. So how do we reconcile these two things? If your toes freeze, you don't enjoy that very much. Some of these things can survive freezing completely. So from an applied perspective, the question really is, will these things survive in Canada, or where in Canada will they survive? From a a basic science perspective, there's just the general question of like, how do they thrive in these places? What are the strategies? How do they evolve? But also there's a kind of a mechanisms kind of question, which is how do they deal with these really, really kind of robust stresses of being frozen or thawed or having, so emerald ash borer, for example, has 4 molar glycerol in its blood during the winter, and that's too viscous to pipette. So how do they deal with these super high concentrations of stuff? So there's always like questions about how biochemistry is working, how metabolism is working, how physiology is working in at low temperatures. And and some of it comes back to this question with the flies, right? Is like, why can I freeze goldenrod gall flies kind of any day of the week during the winter and have them survive, but I can't freeze Drosophila when I want to go on vacation?
Right. And so that raises a very interesting question. And, I think we see a lot in science fiction movies where they will use freezing or cryopreservation for things like space travel. If you need to travel 300, light years, you can just freeze the people and thaw them out when they're ready. Yep. And now it seems like the work that you're doing, however, based on that, are we going to be relying on fruit flies for future space exploration? Do you think they're going to be the best space explorers?
Cockroaches, I think.
Oh, cockroaches? Okay, yeah.
Because
they can take the radiation as well, I guess, right?
Take the radiation, you can store them, they're pretty flat, so you can basically like stack them. Not fussy eaters. So I would expect like very, wide, flat interstellar spacecraft.
Very aerodynamic. Yeah.
Very aerodynamic. Yeah.
It looked like a flying space saucer. Actually, we're probably already being visited occasionally by cockroach space travelers.
Yeah, those are just like big multi-story cockroach.
Where what's the gap between now, like being able to freeze an insect to being able to cryopreserve a human?
Well, I think the first thing that we should say is we can't yet take an insect, Joe insect off the street and freeze it, right? There's only some species that can survive it. Even with Drosophila, with my colleague Vlad's work, it's really kind of hit and miss, and it's not as effective as they might have hoped. And so they did this by feeding them massive quantities of the free amino acid proline. Okay. So we don't, and right down to basic things. So for example, we take these freeze-tolerant insects, we freeze them, we thaw them out, and they still metabolize, right? But even like an isolated mitochondrion, you can't freeze an isolated mammalian mitochondrion and thaw it out and have it work.
Huh.
So we're a long way from that. And so the argument, my colleagues that work on hibernation, on mammalian hibernation, are like, oh, we're going to figure this out, which is also good. But they're sort of in the same situation, really. They can study animals that can hibernate.
Yeah.
But they can't take an animal that can't hibernate and make it hibernate based on all the things they knew. So that's, and that, I guess, brings us into the kind of the paper, which we're probably not going to have time to discuss, that we were going to talk about, but a direction that the lab is taking, which is that we're working on this cricket, Gryllus velitus, the Springfield cricket. So it's a standard black field cricket that you hear calling, guess what, in the spring. And they overwinter as Leighton star nymphs, so basically like little mini crickets, and they can survive freezing. And so where I'm sort of focusing a lot of the NSERC related stuff right now is kind of like, let's like actually systematically explore this. Let's figure out what is it that makes this cricket survive freezing and this other one not survive freezing. The advantages for being able to do this is we can rear them in numbers in the lab. We can make them freeze tolerant. So we can take 2 crickets, this one is freeze tolerant, her sister is not. And they're big enough that we can kind of get at individual tissues and individual cells and things like that. So it's relatively easy to dissect them and say, rather than say grinding up a whole animal and measuring its transcriptome, we can study a specific tissue or a specific cell type within a tissue. Disadvantages, they're still a bit slow. They take a few weeks to acclimate, they take a few weeks to grow and things, but I think that it's really a model system that we can really kind of make some progress with. So, and where we're at the moment is that we can manipulate, for example, their hemolymph, their blood composition. We're working on a protocol for RNAi knockdown of genes that we're interested in knocking down, that sort of thing. So those sorts of things are coming about. Every now and then, lots of people want to work on CRISPR, right? So people will call up and say, like, I want to work on CRISPR on your things. And I maintain that we're a decade or maybe a generation away from knowing what CRISPR we want to do in these animals.
Yeah, okay.
But
we still have quite a bit to learn about the functions of different genes and what you'd want to actually splice in or splice out.
Absolutely. And what's apparent? So, for example, we can take some of these standard cryoprotectants, glycerol and trehalose and proline, and inject them into the animals and make them more freeze tolerant if they're already freeze tolerant. This was work done by Yantina Toxopeus, who is a PhD student in my lab. She's actually about to start a faculty position at St. Francis Xavier University in Nova Scotia. But we can't take an animal that wasn't freeze tolerant and inject this stuff and make them freeze tolerant.
Interesting.
So there's other stuff going on. There's stuff associated with protecting the cytoskeleton. There's stuff associated with changing the way that the organisms like phospholipid membranes respond to freezing. There's got to be protective stuff, making sure that the mitochondria still work. If you've frozen and therefore dehydrated most of your cells, how do you then get all of the iron balance and stuff working okay in your nervous system?
I mean, there's so much going on when an insect is freeze tolerant. There's so many processes going on simultaneously, and so you have to figure out all those mechanistic parts in order to make it. I know.
So it's like really like kind of the world is our oyster in terms of like where to go with this. We had a little session just in a lab meeting where we were like, let's like write down all the experiments we think we should do, and we fill the whiteboard very easily. And it's actually that one of the things I do is I talk at conferences, sort of saying like, hey, here's this model, like it's this cool thing that we can study this. Let me know if you want some, and things, trying to build it up as a model. And one of my slides sort of says like, I know that there are a bazillion other experiments we could be doing on this. Please don't ask me because we've probably thought of them. We just haven't gotten around to it.
It's very easy to like raise your hand and ask a question. Have you guys thought of doing yada yada after I talk? Yeah.
And it's like, because there's one of me.
Yeah.
There's one of me. I'm like my three students.
I want to briefly just ask, what are some skills or personal qualities or general philosophies in life that's made you successful?
Am I successful? Apparently, sometimes. I'm perpetually astonished by this when I go to conferences, and I'm apparently the old dude that like remembers all of the names of people that cite that are long retired and that sort of thing. So for me, and this is, and to some extent I've fallen on my feet in a system that rewards the kind of person I am in terms of being able to just kind of follow my nose a little bit with NSERC funding and things like that. I don't think I would have done as well in the US system where you're really kind of singing for your supper, writing these kind of super focused grants and things. Like I don't think I would have ever have written a good NSF grant, to be honest. I probably would have eventually gotten funded because a program officer somewhere felt sorry for me. So for me, I think the key thing, so there's a couple of things. So I think figuring out what it is that you enjoy doing. So in my case, the world is probably a safer place because I'm not in the lab every day with my sleeves rolled up doing science. Because I like writing, I like helping people figure out how to write, I like helping people think through their projects. So I'm intellectually extremely hands-on in that I really want to know everything that's going on, but I'm practically quite hands-off. And I'm comfortable with that. I have colleagues that would just hate that. They just hate it, right? I'm fairly extroverted and gregarious, which helps, but is not a prerequisite. I want to kind of make that clear. But where it does help is that I end up knowing a lot of people, hanging out with a bunch of people at conferences and things. And that has allowed me to be a little bit of a kind of a nexus for the community that I kind of help people get together, act as a little bit of a matchmaker for students and projects and experts and things like that. And so that I think has helped me become known possibly above the pay scale of the quality of my publications. And I think the other and the other thing is like enjoying writing in all its sort of facets, right? Because really we're professional writers who happen to do a bit of science on the side.
That's what I'm starting to learn anyway. Yeah.
Yeah. So from my perspective, and I think that goes as far as even writing grants. My advice to my students when they get jobs and they're like writing so many grants, all of them fail, this is miserable, I hate writing grants, is that the beauty about writing a grant is that everything is possible at the point where you write the grant, right? You don't have any annoying like data in the way. You haven't had you haven't had a student quit and go off and join the circus midway through the project. You haven't you haven't had a pandemic come in and kill your field season.
Gosh, yeah.
And so I think you have to treat grant writing as an intellectual and collegial pursuit. I have a grant right now with a colleague, Thomas Buckley, in New Zealand, and that grant got rejected 6 times between the previous grant and the one that we're on now, right?
Wow.
And at one point, we were just kind of like chatting on Skype, and we're like, why are we doing this again? And we concluded the reason was that it was actually really fun to spend 3 weeks a year tossing ideas around and coming up with some like wild thing that we thought would answer some important question. And whether or not you get the money is, and I'm speaking from the position of privilege of having a job, having a full-time permanent position where I'm not required to produce any of my salary from grant funding.
Right.
But, it's sort of in some ways like it's only one step removed from what professional philosophers do for a living, right? It's kind of like throw ideas around and stuff. And so it's an immense privilege to be able to do that and kind of, I think, enjoy it. So those are my things. I think be curious, be nice to people as much as you can. Worry a little bit about what you're doing. One of our philosophies is that just everything that leaves the lab is in the best shape we can possibly make it. It may get to the referees and they'll tell us it's crap, but it's not because we didn't put everything into it. And be happy with whatever successes you get. Like, it's just as exciting. I am just as excited about a short note in the Canadian Entomologist as I am about, a paper that I was going to say as a paper in science, but that has never happened.
Yeah, that's a bit of a gap.
Yeah, as I am with a paper in a solid journal where we do publish, because we put our best effort into it, and it was we thought it was worth doing, and we think that other people should hear about it. So,
I want to thank you so much for your time. I know you're very busy, and this is just another Zoom meeting you have to sit in on. So I really do appreciate it.
Okay, cool. Well, thanks for thinking of me. This is a really cool endeavor.
Yeah, thank you so much. And
cool.
we'll keep in touch, and the best to you.
Cheers. See you later.
All right, take care. Yeah, bye.